Next, we're going to be talking about the ways that proteins are modified once they're imported into the rough endoplasmic reticulum or into other organelles. Proteins are modified in some combination of ways, including cleavage, splicing, chemical modification, glycosylation, and folding. Let's start by talking about these two processes known as cleavage and splicing. On this slide up at the top, you're just looking at a ribosome, shown in this green color, traveling along the length of mRNA, shown in this aqua blue color. tRNAs are shown in yellow, and they're coming in and interacting with codons, bringing amino acids, shown in pink. And of course, the chain of amino acids is growing. That's the polypeptide. Remember that newly synthesized polypeptides are in the primary structure. They are literally just a long string of amino acids that have been joined together with peptide bonds. The first thing that happens to those polypeptides is that pieces and parts of them are actually going to be removed. Remember, we said that in the example of co-translational import, the very first thing that gets removed is the signal sequence. There are other amino acids that will be removed too, including, for example, the methionine amino acid that occupies the position where the start codon was in the mRNA. Every single polypeptide has methionine as its very first amino acid, at least in the protein coding region. So methionine is in the first position, the number one position over at that N terminus of the polypeptide. This methionine is going to get removed from every protein. We refer to this as cleavage. Now, the word cleavage tells us that an amino acid or a group of amino acids are sitting at one end of the polypeptide or the other and are being removed. The word splicing is different. Splicing refers to the removal of amino acids that are sitting inside the internal regions of the polypeptide, somewhere or another. So cleavage is removing amino acids that are at either end, and splicing is removing amino acids that are somewhere in the middle of the polypeptide. So all polypeptides are going to have that methionine cleaved off. Polypeptides that are destined to become functional enzymes often have really large sections of amino acids cleaved off during this modification process. And that removal of all those amino acids is going to be part of what helps that enzyme become activated. Of course, there are other polypeptides that get spliced. They're going to have regions in the internal area of the polypeptide removed. A good example of a polypeptide that gets spliced is the one that will become the protein called insulin. Large areas of amino acids in the internal region of the insulin polypeptide are going to be removed as insulin gets processed. Now, when we look at splicing, it's very analogous to the kind of splicing that happens to messenger RNA. Remember, in eukaryotic cells, part of the processing that occurs in the messenger RNA molecule involves cutting out the introns and bringing the exons together. Well, in proteins, we refer to in-teens and X-teens inside the sequence of amino acids. The in-teens will be spliced out, and the X-teens will be brought together and joined to make a more mature polypeptide. So here's a quick question for us to consider. All of this talk about cleaving and splicing amino acids off of polypeptides reminds us that there are enzymes that do this work, just as there are enzymes that build peptide bonds. There are also enzymes that break peptide bonds. Do you know of the name of any enzyme family that's capable of breaking a peptide bond? Any ideas? I actually mentioned one of these families earlier in the lecture. Remember when we looked at the translocon in the membrane of the rough endoplasmic reticulum? And we saw that there's a region of the translocon that is an enzyme. That enzyme is called the signal peptidase. So yes, peptidases are a family of enzymes that are capable of breaking peptide bonds. There is another large family of enzymes called proteases that are also capable of breaking peptide bonds. Proteases and peptidases. So cleavage and splicing are common modifications that are made to newly formed polypeptides. Next on our list of modifications are what we just refer to as chemical modifications. And by chemical, we're referring to small molecules that are sometimes added on to one or more of the amino acids in the polypeptide. I've listed here the three most common small molecules that are added on to polypeptides as chemical modifications. There are methyl groups added on through a process called methylation, acetyl groups through acetylation, and phosphate groups through phosphorylation. Remember, any time a small molecule like one of these gets added on to a polypeptide, it's going to influence the way that that polypeptide ultimately folds up. Next on the list of modifications that are commonly performed on polypeptides is the process known as glycosylation. It's very common for proteins to get modified with small amounts of carbohydrate. Carbohydrate groups, individual sugars, get added on to proteins through this process. Remember that anytime you add carbohydrate to a protein, you have now created what's called a glycoprotein. Finally, the last type of modification that gets done to a young polypeptide is a critical process known as folding. It's folding, of course, that will help determine the ultimate three-dimensional shape, or what we call tertiary structure, for each protein. On this slide, we're looking at how an individual amino acid can be modified through the addition of these small chemical groups. In the drawing, on the left-hand side, you can see the amino acid lysine. Now, you're not looking at the entire amino acid here, you're just looking at the R group. So there is a central carbon that you can't see, and of course that central carbon is attached to an amino group and a carboxyl group and a single hydrogen atom. This structure that's drawn is the R group for lysine. And remember, the R group is different for every amino acid. When it comes to lysine, most of the R group is composed of carbons and hydrogens. Remember, anytime we draw a chemical structure like this with a jagged black line, at every point that represents a carbon atom. You can see that the carbons are bonded to each other covalently. What you can't see drawn in are additional hydrogen atoms that are bound to the carbons. And then, of course, at one end over here, we can see an amino group. Now, when this lysine is modified, there are a couple of ways that it can be adjusted through the addition of small molecules. Lysine can go through acetylation and have an acetyl group added on to the end of the R group in the area of where the amino group normally sits. So we would now refer to this version of lysine as acetyllysine. Lysine is also commonly methylated when it's modified. This is the version of lysine that is commonly produced through methylation. This is actually a trimethyl lysine. And you can see that, again, at the end of the R group, in the area of where there used to be an amino group, we now have 3 methyl groups added on. So depending on the polypeptide and the eventual protein that's going to be made, cells can modify individual amino acids through the addition of these types of small molecules, chemical modification. So when these polypeptides are produced through translation, some of them are going to go through the process of co-translational import, depending upon where their ultimate final destination will be in the cell. And after the polypeptide is drawn into the lumen of the rough endoplasmic reticulum, it undergoes a lot of processing, a lot of modification. We've talked about cleavage and splicing. We've talked about the chemical modifications that are made, the glycosylation process that happens sometimes. And last on our list is folding. Remember, every polypeptide has to be folded into some three-dimensional shape. Each protein has its own unique sequence of amino acids in it, and that sequence of amino acids is going to determine how that protein ultimately is folded. It's interesting to note that in our list of modifications, most of the proteins that are targeted to the rough endoplasmic reticulum, so proteins that are eventually going to reside in the rough endoplasmic reticulum, they're often glycoproteins. So if a protein's ultimate destination, in other words, is going to be in the rough endoplasmic reticulum, it's oftentimes a glycoprotein. Carbohydrate side chains will get added to the protein in the lumen of the rough endoplasmic reticulum. Sometimes those glycoproteins will leave the RER temporarily, get packaged into transport vesicles, and go on to the Golgi apparatus. And they do that so that they can have further carbohydrate modifications added to them. Now, remember, any protein that's going to travel to the Golgi apparatus is going to come in through the cis face and then travel through the layers of the Golgi apparatus. That's where further glycosylation can happen, or carbohydrates that have been added to that protein can be modified. Eventually, that protein will get to the trans face of the Golgi apparatus. It'll get packed into a vesicle and it will be sent to its destination. And of course, sometimes that destination is back to the rough endoplasmic reticulum. But let's move forward now and talk about the process of folding. Remember, polypeptides, chains of amino acids, are going to go through folding as a step-by-step process. We said that the first thing that's going to happen is alpha helices and beta pleated sheets are going to form. And then those alpha helices and beta sheets will interact with each other, and you will get some sort of a complex three-dimensional shape. Now, it's probably not a surprise to you that because of the size of most polypeptides, remember, hundreds to thousands of amino acids, because of the size of those molecules, it's possible to fold them up in multiple different ways. It's possible to create hydrogen bond patterns and van der Waals interaction patterns in different ways and still end up with a folded protein. However, there's only one shape that that protein needs to get into in order to serve the function that it has in the cell. So it's going to be critical that folding occur in a managed way. You can't let most polypeptides just spontaneously fold if you're a cell, because they may or may not fold into the right shape for the function of that protein. For that reason, cells invest a lot of materials and a lot of energy into the folding of proteins. The big player, the big regulator of protein folding is another type of protein called a chaperone protein. In different source materials, you will hear these referred to sometimes as molecular chaperones, sometimes just as chaperones, and sometimes as chaperone proteins. Some version of these three words to describe these important molecules that guide protein folding. Now, interestingly enough, these chaperone proteins are referred to, at least in some of the older literature, as heat shock proteins or HSP proteins. They got that name because of the way they were discovered. It turns out that when cells experience what we call a heat shock, including a purposeful heat shock in the laboratory, one of the molecules that can suffer from excessive heat is protein. So when a cell experiences a heat shock, what will happen is the cell will produce more chaperone proteins because there's going to be some damage to existing proteins, and those proteins may need to be refolded after the heat shock. So they were discovered as scientists studied what happens to a cell when it undergoes heat shocking, and they got the name heat shock proteins as a result. The most abundant molecular chaperone is called heat shock protein 70, or HSP70. This particular heat shock protein, or chaperone protein, also goes by a second name, and this second name is the more recent name that was given to it, which is BIP. This stands for binding protein. Now that is about as generic a name as I can imagine giving to a protein, especially one as important as this one. Heat shock protein 70, or BIP as it's also known, is going to be very abundant in the rough endoplasmic reticulum because, of course, a whole lot of proteins are going to travel through the RER during co-translational import. This molecule, this HSP70 molecule, will be in the lumen of the rough endoplasmic reticulum, right near the translocon. It sort of lives near the translocon, near to where the polypeptide is going to first come in to the rough endoplasmic reticulum. Now, when that polypeptide comes into the lumen through the process we've discussed, the heat shock 70 protein is going to bind specifically to some hydrophobic area on that polypeptide. Now, it's going to stick to the polypeptide and then it's going to guide the way it folds. One of the most important things that HSP70 is doing, or BIP is doing, by coming in and binding to that brand new polypeptide, is it is preventing it from starting to fold spontaneously. Remember, that string of amino acids has a lot of chemistry. There are polar amino acids and non-polar amino acids and acidic ones and basic ones, and attractions are going to start to form. The polypeptide is going to begin sort of binding to itself and folding up all by itself with no guidance. So part of what HSP70 is doing by binding to that polypeptide is it's going to prevent it from folding to itself, and it's going to give it time for other chaperone proteins to come in and start the folding process step by step by step. These heat shock proteins, or BIPs, are actually binding to and then releasing from the polypeptide so that the natural attractions can form one at a time in the right sequence. It's important to note that folding requires energy. So there will be some hydrolysis of ATP going on. And essentially what that energy is being used for is to allow the chaperones to bind and then release and then bind and then release in a very ordered sequence so that the polypeptide folds up correctly the first time. Now, it's also important to note that sometimes during the folding process, a step goes wrong and a fold is made that is incorrect. So part of the binding and unbinding that happens with the chaperone proteins is important because you can take a step backwards if you need to and try to unfold something that was folded wrong and refold it. Step by step by step, this great big protein will be put into its correct three-dimensional shape. On this slide, we're looking at a drawing of the membrane in some area of the rough endoplasmic reticulum. We can see the outside cytoplasm and the inside lumen of the RER. You can see a ribosome drawn in pink and a strand of mRNA being translated. We're dealing here with a polypeptide that's going to go through co-translational import into the RER. You can see the signal sequence is still stuck inside the pore. That's what you're seeing in red. Each little circle represents an amino acid. We've gotten to the point here where, at least in this drawing, it looks like the signal has already been cleaved off by the signal peptidase enzyme, which is right here. And the artist is showing us that in the lumen of the RER, binding protein is sitting and ready to attach to this gray polypeptide as it comes through the pore, so that any folding that takes place will be guided folding. Nothing spontaneous is going to be allowed to happen.